Good afternoon, everybody. If you're joining us from around the world, good evening and good morning. Thank you for joining us for another session of Science from Your Sofa. I'm Amy Liebman, President of NTM Info, and I will be your host and moderator today. We are very excited today to welcome Dr. Ashley Slagle, who will be presenting on Measures That Matter, a regulatory perspective on patient-reported outcomes in drug development. Ashley Slagle is the principal and founder of Aspen Consulting, where she provides strategic, regulatory, and scientific advice to drug developers on matters related to patient-focused drug development, with a particular focus on endpoints and clinical outcome assessments, or COAs, including patient-reported outcomes, or PROs, to support medical product approval. Dr. Slagle's experience includes over 20 years of health outcomes research, policy analysis, and COA development, implementation, and analysis to evaluate treatment benefit for regulatory purposes. She advises on the selection and development of COAs for use in clinical trials, as well as related study design issues and clinical trial endpoints. She participates in the design, implementation, and analysis of both qualitative and psychometric research studies, instrument modification and development, translation and linguistic validation, and migration of paper instruments to electronic platforms. Dr. Slagle previously served as the scientific coordinator for the FDA CDER Drug Development Tool COA qualification program. While at the FDA, she provided oversight on review and recommendations related to the development and use of COAs, and worked closely with all CDER review divisions to make drug approval and labeling decisions. Dr. Slagle also participated in FDA policy and guidance development and legislative activities. She received her Master of Science and PhD in Pharmaceutical Health Services Research from the University of Maryland at Baltimore. Today, during the presentation, you can ask questions by typing them into the Q&A box. When you are done typing your question, you hit send and the questions will go into the queue. At the end of the presentation, I will be reading the questions aloud. Don't forget to join us for our upcoming webinars also in September. On Friday, this Friday, the 24th, we have Medical Management of GERD with Dr. Gwen Hewitt from National Jewish. And on Sunday, September 26th, we have the next session in our series on emotional well-being with Dr. Devin Smith. She'll be presenting on changing thought patterns to decrease emotional distress. I'd like to take a moment to acknowledge the ongoing support of several companies, including International Biophysics, Insmed, Maxor, and Paratech. Without the support of patrons like these, we could not continue to do this kind of programming that we bring to you year-round. We'd like to thank them for their support. Don't forget to find us on Instagram, Facebook, Twitter, and YouTube. And you can find all our videos on YouTube as well. So you can follow us on social media, and you can learn more and sign up for our email newsletter at ntminfo.org. And now I would like to turn this over to Dr. Slagle. Thank you for joining us today, and we look forward to hearing what you have to say.
Thanks, Amy. Let me just share my screen here. Great. Well, thanks for the invitation to be here today. You know, I'm really passionate about patient-focused drug development, and a cornerstone of that is measuring what matters to patients in clinical trials, so that when products are approved, there's the information available that's relevant to patients and their providers, really to make informed treatment decisions. Often we use patient-reported outcome measures to provide this information. But I've found during my more than 2 decades in the pharmaceutical industry that unfortunately, often people don't fully appreciate how important it is and how challenging it can be to get these measures right for use in clinical trials. It's not as simple as pulling a questionnaire off the shelf to put into a trial. Really, a lot of work has to go into developing or selecting these measures so that we can trust the information that comes out of them and they're truly useful in future decision making. So these measures used in trials obviously need to be acceptable to regulators. So I do want to share today some really regulatory perspectives, but also just good measurement development principles that I hope can shed some light and help understand help us all understand a little bit better how to use PRO measures appropriately in clinical trials. So I'm sorry to start out with definitions, probably the most boring part of this talk, but sometimes I see people talking past each other because we use these words interchangeably and they're not meant to be interchangeable. So an outcome assessment and endpoint are two different things. The outcome assessment is the actual tool that we use to collect the data. So this is the questionnaire, for example, that we put into a clinical trial. Sometimes, just to be confusing, we call these different things: outcome assessment, tool, instrument, questionnaire. But these are different from an endpoint, which is the variable that we use in a trial. So it's statistically analyzed and it tells us something about how the treatment is working. An endpoint is analyzed using a score from the outcome assessment or questionnaire. So obviously, as you can tell, outcome assessment, also known as questionnaire instrument, and endpoint are not synonymous. So this is just an example to try to make that point a little bit further. So say we're interested in looking at respiratory symptoms of NTM lung infection. So that's the overarching outcome we're interested in. In the trial, we might use something like the QoL-B questionnaire for the outcome assessment. And while we may include the full questionnaire in the trial, we're most interested in the portion of it that assesses respiratory symptoms, or in measurement jargon, we call this the respiratory symptoms domain. So when we consider the endpoint then, that builds on the use of the QoL-B questionnaire, but the endpoint then is further described to consider how we will analyze the score of that domain. So for example, we might decide to measure and analyze the mean change from baseline on the QoL-B respiratory symptoms domain compared across treatment arms at, say, month 7. So you can see there's a lot more information that goes into thinking about an endpoint beyond the questionnaire. Of course, because I'm a former regulator and we learn to caveat everything, I do want to note this is just an example to illustrate the differences in these terms, and it's not my intention here or throughout this webinar to endorse any particular instrument or endpoint for use in clinical trials. So another term that comes up a lot is validated. I've grown to really dislike this term because I think, unfortunately, it's led to some poor decision-making in the past. So often, existing questionnaires are developed in clinical practice, not specifically for regulatory use. And sometimes they're developed even without any patient input. These are often described in the literature as validated because they have some psychometric evidence from their use in the clinic or maybe in an epidemiologic study. But the important point here is that no instrument is validated for all purposes. Often, these existing instruments are not, in fact, appropriate for use in clinical trials to evaluate treatment benefit. I recall one of my first examples of this as a new reviewer at FDA some years ago. There was a symptoms measure that was proposed for use in a clinical trial. It was described by the literature as validated. It had been developed and used successfully in clinical practice to help clinicians evaluate and diagnose patients and to help gauge their clinical status over time. But when you inspect the instrument, the questionnaire itself, there were questions that were included that, while important to clinicians to use as a tool in clinical practice, the questions would not be useful in a clinical trial. So for example, one of the questions asked at what age the person's seizures started, and a longer time since seizure onset was a worse score to indicate that the patient was likely in a more severe state of the disease. But clearly in a treatment trial, we're looking for change in people's clinical status to see if a drug product is working. Obviously, the age at which seizures started is not going to be changed by even a successful treatment. So questions like this in a measure that are intended to be used in clinical trials are going to bias towards the null. That means it's going to make it harder to show treatment benefit even when it exists using that instrument score. So this is a pretty obvious example that an instrument validated for one purpose was not indeed fit for purpose of showing treatment benefit in a clinical trial. We also might see examples where different questionnaires might be needed for different subpopulations of patients. So an instrument validated in one subpopulation of a condition may not work well in another subpopulation. So for example, there might be questions that evaluate how a drug is working quite well in a subset of people who have mild or moderate disease, but that same questionnaire may not work well in patients who have more severe disease. So it's always very important to ensure that the questions in the questionnaire actually match the exact target population that will be in the clinical trials. So when we think about an outcome measure for clinical trials, it's important to evaluate, or if needed, generate the evidence that the instrument is fit for purpose for your clinical context. So I always find myself on guard if someone describes the tool as validated. It's important to question that carefully. Validated for what? Is it fit for purpose for the particular clinical program that it's going to be used in? The instrument might very well be acceptable. I've just heard the term validated used incorrectly so frequently. And then when it's relied upon without questioning, it has led to problems with trial outcomes and endpoints. So I like this example that I think sort of helps drive the point home, where a validated questionnaire may not be appropriate for all people with a given condition. So this is another real example I saw when I was a reviewer at the FDA. This is where a questionnaire may work well for one severity level, but not so well in a different severity level. So this is just one example of a question that was in the questionnaire. Of course, this was a questionnaire that was described by the sponsor as a validated measure of physical function. It was intended for use in a very severely ill patient population who was largely bedridden and minimal physical activity was very difficult. The sponsor was developing a product that they hoped would improve patients' physical functioning, which was an important goal of treatment, clearly. But upon inspection of the questionnaire, there were questions like, do you have trouble running to the bus? So clearly this question is not appropriate for this patient population. And even with a successful treatment, the intention was only that there would be modest physical function improvements. So this question would never be relevant. This question would show no movement, even with a successful treatment. Again, this is an obvious example of a question that's not appropriate, that can mask a treatment effect if it exists. There are, of course, other problems with instruments that are not so obvious. And so that's why it's really important to do all the right research to ensure that a measure is indeed fit for purpose for a clinical trial. So just to sort of recap, the purpose of an outcome assessment in a clinical trial is to be able to analyze it, to determine whether that product provides treatment benefit to patients. And then a conclusion of treatment benefit is described in the approved product's labeling or prescribing information. So of course, then one of the most important aspects of drug development is how treatment benefit is measured. So ultimately in trials, we are seeking to evaluate that treatment benefit. That's also sometimes called clinical benefit. So those terms are interchangeable. I happen to use treatment benefit, but a lot of other FDAers and ex-FDAers use clinical benefit. So this is that a drug has some positive impact on something that's important to people with the condition. And specifically for FDA treatment benefit or clinical benefit, this relies on knowing one or more of the following about a treatment's influence on either survival or how patients feel or function in daily life. This is really critical. So treatment benefit, as defined by FDA, is an influence on survival or how patients feel or function. Of course, FDA then has to weigh those benefits that are demonstrated and quantified in a trial or clinical trials with the known risks of the product in order to make drug approval and labeling decisions. So there are different types of outcome assessments that can be used to evaluate treatment benefit. Of course, there's survival, how long a person lives. However, in some contexts, survival may not be an appropriate endpoint in a trial, either because the condition does not impact survival or because the trial would need to be prohibitively large or long in order to detect survival benefits. So we often find that we're turning to other types of assessments to support endpoints in clinical trials. So there are clinical outcome assessments and surrogates. And what really separates clinical outcome assessments from surrogates, which are typically biomarkers, is that clinical outcome assessments, or COAs, rely on some human judgment, motivation, participation. A person is reporting on something or doing something and being observed. I'm going to share a little bit more on COAs in the coming slides, but I do want to spend a minute talking about surrogates here because they're often used in anti-infective research. When we think about surrogates, these are things that are meant to be a substitute assessment for how a patient feels, functions, or survives. And there are two main types that are used in regulatory decision-making. Those that are established with a lot of evidence over many years of research that we know are good proxies for how patients feel, function, or survive. Blood pressure is a good example here. Then there are surrogates that we believe are reasonably likely to predict benefit, but we don't yet have the full set of evidence to provide absolute confidence that that's the case. I think it's helpful to look at this continuum of evidence of treatment benefit. So on the left, we have those things that are, we're still a little bit unsure about how closely they represent true treatment benefit. Again, that is how a patient feels, functions, or survives. On the far right, we have those things that provide more direct evidence and thus greater confidence that these actually reflect how a person feels, functions, or survives. These are things like direct measures of patient symptoms or patient measures of activities of daily living. In some cases, while they're still not direct measures, we do have more confidence of these established surrogates that we know predict survival or other aspects of treatment benefit. So this idea of direct and indirect evidence of treatment benefit has really very serious regulatory implications for approval. In fact, FDA has different types of approvals based on the type of assessment or endpoint used to provide evidence that the treatment is working. So there's traditional approval. That's what most products receive. And it relies on those direct measures of treatment benefit or a validated surrogate. Then there is accelerated approval. This relies on one of those indirect measures of treatment benefit. So a surrogate that is reasonably likely to predict benefit. This has an advantage of sometimes providing much faster time to approval. And it's helpful in those conditions where relying on a direct measure could slow drug development in an unacceptable way. However, of course, the drawback clearly is that we don't know for all certainty whether the treatment provides true clinical benefit. So with accelerated approval then, drug sponsors must complete post-marketing studies, phase 4 studies, with the aim to confirm that there is actually benefit on how patients feel, function, or survive. So this was the case in the ARIKAYCE approval. So this product was approved under the accelerated approval pathway. The primary endpoint was sputum culture conversion, and advisory committee members who are outside experts who advise FDA indicated that the product was reasonably likely to predict benefit. But these approvals are not at all easy for FDA. They're thinking about where do we draw the line on how reasonably likely something is to provide benefit? So following this approval, FDA convened a meeting to discuss drug development for NTM, which included an in-depth discussion of outcome measures, specifically patient-reported outcome measures that could be used in future studies to directly measure treatment benefit, in addition to the surrogate endpoints that are used. So patient-reported outcome measures are often the most direct way. We're asking patients just directly, how are they feeling or functioning in trials? But we can also measure treatment benefit using other clinical outcome assessment types. So I included this slide really for comprehensiveness. If patients can't report for themselves, say in pediatric studies, we might use an observer-reported outcome, asking parents or guardians to report on the measures. Of course, we can have clinicians making clinical observations and reporting on a clinician-reported outcome measure. And then sometimes we can use performance outcomes. These are things like the 6-minute walk test, where we ask people to complete some sort of defined performance task, and then they're scored in some way doing that task. So you can see we can measure treatment benefit in many different ways. I'm going to focus on PRO measures for the rest of the discussion, because I think they're the most relevant, especially right now in NTM lung disease, given that this population can report for themselves, and there are clear, relevant, important symptoms that are part of the NTM lung disease experience that are best reported from the patient perspective. And then also because there's an ongoing focus by drug and instrument developers to identify or modify or develop a fit-for-purpose NTM disease PRO measure or measures that can be used directly to measure treatment benefit in clinical trials. So again, as a former regulator, I can't help myself but from sharing the actual regulations. But I do think this is important to remember. From the regulatory perspective, it's necessary that treatment benefit is documented with substantial evidence from adequate and well-controlled clinical trials. And the regulations also specifically indicate that the methods of assessment of a subject's response should be, quote, well-defined and reliable. So this is really important. It means that well-defined and reliable become the key criteria by which FDA judges outcome assessments, including patient-reported outcome assessments, to evaluate evidence of treatment benefit. So when is a COA or a PRO assessment well-defined and reliable and appropriate for use in adequate and well-controlled studies? Well, when we're measuring the right thing in the right way in that particular patient population, and the score that quantifies that thing that we're measuring does so validly and reliably, so that we can believe any of the effects seen on that outcome assessment. And we can interpret that as clear treatment benefit. So from FDA's perspective, we refer to the PRO guidance, FDA PRO guidance, that describes good measurement principles that might be considered to evaluate whether a measurement is well-defined and reliable. So this guidance was developed for patient-reported outcomes, but many of the principles are appropriate to apply to any COA type. And this guidance provides really an optimal approach to PRO development, but FDA increasingly understands that flexibility and sometimes creativity are needed in order to both meet regulatory standards, but also the practical demands of drug development. And in fact, this guidance is expected to be updated with a new guidance that's part of FDA's patient-focused drug development initiative. And while we expect it will maintain the rigorous evidentiary standards for PRO and other COA evidence, we hope that it will provide some more practical approaches and examples really to help drug and instrument developers. In the PRO guidance, and expected in the forthcoming patient-focused drug development guidance, we always rely on long-standing good measurement principles. And these include documenting different psychometric properties of the PRO measures intended for use in a trial. And these properties include content validity, construct validity, reliability, ability to detect change, and then some information to help interpret what is meaningful change on that instrument. So often when people think of validated, they think of the psychometric properties that are listed here in black font. Unfortunately, though, the two key properties that really are of critical importance to FDA decision-making are sometimes overlooked, and that is content validity and the information to support interpretation of meaningful change. So content validity evidence really provides confidence that we're measuring the right things in that population, that the patients reporting on the questionnaire understand the questions and are able to respond in the way intended. It gives us confidence that when we combine all of the questions in the questionnaire into a single score, that score is sufficiently comprehensive and we understand what that score truly represents. So regulators put a big emphasis on content validity because they need to ensure when they see a score change that they understand what it means, and we can describe that score change in product labeling in terms of what was actually measured, what is the true treatment benefit in a way that's not potentially false or misleading. When thinking about content validity, FDA relies on a conceptual framework. This is just a way to look at how the questions in a questionnaire fit together into a score or a set of scores that might be used as different endpoints. So this is an example of a hypothetical conceptual framework for a lung disease questionnaire that would have been developed by talking with patients to understand what are the important symptoms of their condition. Again, for illustrative purposes only, I'm not making a recommendation on what a conceptual framework for an NTM lung disease instrument should be. But we see here in this hypothetical example that there are three possible scores: a respiratory symptom score, which is comprised of questions about coughing, wheezing, shortness of breath; a non-respiratory symptom score, which is comprised of questions about body pain, fatigue, and chills; and then a total symptom score that combines all of these symptoms into one grand total score. So when FDA evaluates content validity, they need to be sure that each of the scores here are comprised of questions that are sufficiently comprehensive of the patient experience. So meaning that the questionnaire and the scores do not omit any important symptom that's relevant to the people with this condition. So here's the same example, but notice that some of the symptom questions are missing. Here we see that the respiratory symptoms domain does not include the cough symptom, and fatigue and chills have been omitted from the non-respiratory symptoms domain. This could represent a problem for regulators. So imagine if the score of the respiratory domain here, based on wheezing and shortness of breath, improved with treatment. But we would have no idea what happened with cough during this trial because this important symptom was omitted from the questionnaire and the score. Therefore, FDA wouldn't be able to say that respiratory symptoms improved because they have no idea what happened with cough. It could have actually gotten worse. We just don't know. So if they approved a product and stated that it treated respiratory symptoms, you can see how this could be a potentially false or misleading claim. So this is another example of why it's so important to do the research, document what are the important and relevant symptoms, and measure those appropriately in a clinical trial. So construct validity is determined by evidence that relationships between the PRO assessment under study, that its scores relate to other assessments in a way we would expect. So for example, with convergent validity, which is a type of construct validity, we look at the correlations between our PRO instrument under study and other similar measures. So this is sort of a check to make sure that the scores correlate. If they don't, it gives us an indication that either our new PRO assessment or the other instrument or other questionnaire are not assessing exactly what we think is being assessed. So maybe there are some bad questions that are not working in the way we intended. So this really gives us a heads up that we need to take a careful look again at the questions in the questionnaires. Of course, generally of content validity, which is based largely on patient interviews and pilot testing the questionnaires with patients in a qualitative setting, if that content validity work has been strong, then we often see that construct validity assessments are just more confirmatory. Again, speaking to the importance of content validity evidence and engaging in patient interviews or focus groups to build that evidence. Discriminant validity just tells us that measures we would not expect to be related to our PRO measure are indeed not correlated strongly. So for example, this would be like, we wouldn't expect IQ and physical function to be highly correlated. And then known groups validity is where we look at different subpopulations with known clinical differences and ensure that the scores in the different groups on the PRO assessment are indeed different in the way we would expect. So we may look at those PRO scores in a group who has very severe disease, maybe as indicated by a clinical exam, versus those who have mild disease based on that clinical exam. Doing the research to evaluate construct validity requires a large sample of people. So it's often work that's done in early phase trials and then confirmed in later phase trials. This kind of research requires then that we put other measures into the trial in order to look at correlations between our PRO assessment of interest and those other measures. So this is why sometimes in trials, people might feel like they're having to answer a lot of redundant questions or very similar questions. It's not that the researchers are trying to trick the trial participants. It is really to be able to provide that needed psychometric evidence and document that the important PRO assessment that you're studying that will be used as a future endpoint, that it is indeed fit for purpose. In addition to evaluating validity, which is really another term for accuracy, we also examine the reliability of an outcome measure. So where validity is accuracy, reliability is consistency. With PRO measures, we look at test-retest reliability. So when a person's clinical status does not change, we should see the PRO assessment score also does not change. If it does, then we worry that the measure is unreliable, and any score change we see in a treatment trial, we have to worry, was it because the treatment is working or because the measure is unreliable, and this is just noise in the instrument. Also, unreliable instruments have a tendency, again, to bias towards the null, which means it makes it harder to show differences between arms. And so you may have an effective treatment, but if you have an unreliable instrument, you may not be able to show that treatment using that instrument. So obviously, we want a reliable measure. And we also look at internal consistency. This is just to examine whether questions that we combine together into a single score are similar enough with each other to put together into that single score. So also critically important for our measures in clinical trials, of course, is the ability to detect change when there is change. So we can examine this by correlating change scores on our PRO measure with change scores on other similar measures. Again, this has the result of us needing to put those additional but similar measures into trials. So again, what seems like redundancy in questions does have a rationale. So, and it's really important to have good data from trial participants across all of these measures so that we can evaluate the PRO measure under study. So I've covered most of this slide already, describing the type of research that's done to document the different types of measurement or psychometric properties. But the point I want to make here is that there really needs to be a balance between putting enough other measures in a trial to be able to do the needed research, but also balancing that against patient burden. Of course, if we put too many measures in, we risk that it exhausts people, that they become tired of completing the measures or annoyed when they have to complete so many similar measures or complete them so frequently. And then the data may suffer. So we may have missing data, or we may find that people answer the questions, but without giving it much thought because they're just exhausted at the end of this, long battery of assessments. So I think this is a case where really all stakeholders should be working together, sponsors and FDA, by agreeing, to put in the key measures of importance in order to do the research. And then sponsors sharing with trial participants the rationale for all of these measures, why they're seeing duplication and being asked to complete what seems like quite similar questions. And then trial participants, I think, once understanding that the battery of measures is really important to the research objectives beyond efficacy, but also making sure that our PRO measure is fit for purpose. I think having that understanding makes people more willing to be careful and thoughtful when they complete the full set of measures. So I want to just make a final point about outcome assessments and trials. It's really important to remember that available assessments are not all adequate for use as clinical outcome assessments to evaluate treatment benefit in trials. There are assessments that, for example, even while reported by patients and they're useful for very different purposes, they may not be useful in a clinical trial. So I sort of feel like I'm beating a dead horse here, but this is so critically important. And I see this challenge all the time where, measures that have been used maybe for diagnostic purposes or prognostic purposes, or used to select patients for participation in clinical trials, or maybe used for epidemiologic or population studies, and have been used well to understand characteristics or the natural history of a condition, or even to assist in clinical practice decision-making. Those instruments are often not appropriate for use in clinical trials to assess treatment benefit, at least not without some modifications. So just as a few examples, often diagnostic tools are quite broad in order to identify all the patients who have a condition. So for example, a diagnostic tool might include, all of the possible symptoms that a person might have with that condition, even if only a few patients may experience some of those defining symptoms at any one time during the course of that condition. But if we use this diagnostic tool as an outcome assessment, there may be many questions that much of the population isn't experiencing at that time in the clinical trial. And so they wouldn't change with treatment, that there wasn't room to move on the instrument because they weren't experiencing those symptoms to get better. And when there are many items like this on an assessment that don't change with treatment or during treatment in the trial, it makes it harder to see improvement in that total score. So the beneficial effect on a few key symptoms might be lost or obscured by having no effect on the other questions in the questionnaire that are not relevant to many patients' experience. So again, it's really important to match the questionnaire to the experiences the patients who are going to be in that clinical trial are actually having during the clinical trial. Now, sometimes we also see checklists of symptoms used. So these are developed often in clinical practice where a person would respond yes or no to the presence of a symptom. In cases where we expect that people will have meaningful symptom improvement, but not necessarily have complete symptom resolution, I think you can see where this checklist is probably going to miss important treatment benefits. So not having a more a response scale that provides sort of gradations of severity, but only having yes, no as the responses doesn't allow us to see the more granular changes in severity that many patients would still find important. And then sometimes existing measures have scores that combine many different concepts into a single score. So rather than, say, having a respiratory symptoms domain, the score might include all the respiratory symptoms, physical function impacts, and maybe even emotional health concepts. It's not to say that those functional and emotional impacts are not important. Of course they are. But when you combine them into a total score with respiratory symptoms, and we see change on that score, it's not clear what was driving that score change. What has actually improved, what has gotten worse. And then also the functional and emotional impacts. There are a lot of other things besides the treatment itself that can impact those experiences. And so sometimes it may take longer to see change on those. So if we mix all of those things together in a total score, again, we might not see the total score change, making it look like the drug isn't working. But if we look more carefully at just the respiratory symptoms as a concise, specific score, then we may be more likely to see the benefits of a treatment. So you can see there are many things we have to guard against to make sure we're measuring treatment benefit. Trial participants in other instrument development research. Well, of course, one reason is to meet the requirements of FDA. So, we have to ensure that measures are well-defined and reliable, and they can be the basis for drug approval and claims of treatment benefit. But more broadly, we want to ensure that we know what we're measuring, that it's meaningful and relevant to patients, and can be described in a way that is informative and not potentially misleading. And then finally, to be able to detect treatment benefit is, of course, a very important component of clinical trials. When a drug, product fails to show benefit, we really don't want to be left wondering, was it the drug that failed, or did we just use a bad outcome measure and that failed? So that's the end of the presentation, and I'm very happy to take some questions now.
Thank you, Dr. Slagle. This was very informative. I'd like to invite the audience now to ask any questions. This was incredibly helpful, and I think it is very helpful because we do have several clinical trials going on right now that are both using PROs and are looking to develop and validate PROs. So I think it's very important that patients actually learn about this. Somebody actually is asking, does this apply to the COVID research that they're doing? So I don't know if you have any information about whether they're using PROs in COVID research.
Actually, they are, and I have had the privilege to help a couple of sponsors work on different PRO measures. One sponsor work on a PRO measure in a clinical trial in different settings. So yes, it starts, even though we are trying to do things very quickly, it's still important to do things correctly. And so the PRO development, even for COVID, started with patient interviews to understand what are the symptoms that they're experiencing. Also looking to the literature, CDC was putting out information on what symptoms people were experiencing, talking to clinicians who have treated COVID patients, and really using everything we could to understand what are the key symptoms, but largely really relied on the patient interviews because that was the most in detail and informative. And then from those learnings, we developed, and there are multiple measures being developed in the same way, but I'm talking about the one I was involved in. We developed questions for a questionnaire, and then we took that questionnaire back into patients to ask them, did we get it right, basically? Do you understand these questions? Can you report on it? And then those questionnaires were put into trials. And so then we could do the psychometric evaluations, and that's ongoing now. So yeah, the same process is used regardless of indication. There are ways we can speed it up, and we had to do that a little bit in COVID. And we've done that in other indications, but we still need to do the same rigorous evidence generation to make sure that we're using appropriate measures, PRO measures.
Sure. Somebody is asking about, I guess this would relate to harmonization of rules and data across different countries and regions. Somebody's asking, are the regulations the same or similar in the United Kingdom?
It's a great question. When I first started in this industry, they were quite different, especially regarding PROs and what they were willing to put into product labeling, and what they thought about in evaluating the appropriateness of a PRO measure. Over the years, we've seen a little bit more harmonization, alignment. We still see that, especially EMA is more willing to label some of the broader concepts that FDA is not willing to label. So claims like about health-related quality of life, which encompasses a number of concepts, symptoms, physical function, emotional functioning, social functioning. FDA is looking very narrowly, because of their regulations, at treatment benefit. And so they look at symptoms and maybe physical function that's core to the condition. But EMA will, and UK and some of the other European regulators will allow kind of these broader claims, but they still need to know that the instrument is appropriate, and they look at the same measurement properties, the content validity, construct validity, reliability, ability to detect change, and what is meaningful change. So the principles are the same, but the types of concepts they're willing to label is a little broader in Europe than here.
Okay. Somebody's asking, do pharmaceutical companies ever use single subject designs in initial trials, since such a research design would allow you to look at individual symptoms instead of always using group designs?
Oh, that's a very good question. That might be outside of my core expertise. I think there are a lot of different study designs that are being used, and I have seen leadership at FDA be very open to different trial designs. And in some cases, it is seemingly better to look at kind of at the patient level, what is the most important symptom to them? So, well, I can give an example. There's a migraine guidance from FDA where this is not so much about trial design, it's still more about the endpoints. But in migraine, everyone has the excruciating pain, but then they have other symptoms. Some people have photophobia, some have phonophobia, some have nausea, vomiting, but they don't generally have all of those symptoms. And so at the beginning of the trial, you can ask patients, what is your most important symptom in addition to pain? And then that symptom is measured for that patient over time. And a different symptom may be measured for another patient over time. So there are ways to get a little bit more specific at the patient level. This is a little harder to do in some conditions, because migraine is a case where really if photophobia is your core symptom in addition to pain, that usually persists over time. You don't see a lot of change in that. We find in a lot of other indications, unfortunately, it's harder to do that because symptoms may sort of ebb and flow. There's not one single symptom across time that is really bothersome to a patient. Maybe there is, but a lot of times there are other symptoms that are experienced by that patient. So it's a little harder to implement, but certainly there is more, I guess, appetite or consideration by FDA to think about some of these more creative trial designs or endpoint specifications.
Okay, thank you. Before we get to the next question, I'm going to read out a comment that was dropped in. Wonderful presentation, explains the importance of participation in clinical trials. Good to know the importance of feedback from patients. And yes, that is exactly why we've done this presentation today, because it is so important to participate in clinical trials. And it's important for everybody to know that when you answer all of those questionnaires, you answer them over and over again, there's an actual purpose for them. They're really trying to determine that this product that they're developing is going to do something of value for the patient. So here's somebody dropped they said it's not really a question, just a comment, but you might be able to speak to this. Wouldn't it be wonderful if the general population could hear and learn what vast research has been and continues to be done regarding COVID and other vaccines, especially for the vaccine hesitant? Would it be possible? And I don't know, maybe you could speak particularly to, the PROs and maybe the information that's coming out of those PROs might be useful to sort of, generate conversation that might help reach some of the vaccine hesitant population.
Yeah. To me, this is a big challenge in any indication, is that we don't have a great way often to communicate a lot of information to society. FDA has very rigorous requirements. Good research is done, and then they approve a product and they put information in product labeling. And then in our country, the sponsors can, if the product is approved, can promote on that. And so that's one of our key mechanisms to getting information out to people, is through promotion. We publish in medical journals, but it's a subset of very motivated people that are looking in medical journals for their information. And then I'm going to get on a soapbox here. I think the lay press often does a terrible job describing very complicated research. It's a hard task that they have to distill complicated research down into, a digestible format. But I think that's also been a problem. So I don't have any great answers on how to better communicate to people. I think that, to the extent that people can read the actual information in medical journals, that's most helpful. Talk to trusted scientists and their physicians. But gosh, I wish I could solve the problem of vaccine hesitancy through PRO data. I can't. I wish.
Another comment, great presentation. I wish you had taught my graduate school class in research tests and measurements. So, and another question, are any of the current trials showing a particular drug that is exceedingly promising? And I'm not sure if they're asking in the context of NTM or in general. So,
Well, unfortunately, unless or until something's approved, and even if I know about it, I can't share that information because it's all proprietary research. So it's a question I unfortunately can't answer. Sorry.
That's okay, we understand. Normally we find out once the results from the previous phase trial are out. So when we get that information, we certainly put that information out publicly once it's available to the public. Another compliment, thank you, wonderful presentation. I agree, this was wonderful and very helpful, and as you can tell by the questions, certainly provoked a lot of thought, and it's clear that people really got a lot of valuable information from this. So thank you very much.
Sure.
We really appreciate everybody joining us today, and Dr. Slagle, thank you for taking the time. We really appreciate it. Everyone, just a reminder that we have 2 more webinars coming up in September, so don't forget to join us for those. Registration is open, and we hope to see everybody again soon. Thank you very much for joining us, everybody. Everyone have a good week.
Thank you.
Bye.
Take care. Bye-bye.